Assalamualaikum and hi, this is Group 19 presentation. For Group 19 members, we have Kritika, Adam, Arifah and Akmal.
I'll be presenting on the background of our research, which focuses on a high-quality de novo genome assembly of the coconut rhinoceros beetle or resides rhinoceros using nanopore sequencing. Let's begin with what is UGT1A1 is. Well, UGT1A1 is a gene that encodes an enzyme called UDP-glucuronosyl transferase. This enzyme plays a critical role in breaking down and also eliminating substances from the body such as bilirubin and also many types of drugs. Why is this gene important? Well, variations in UGT1A1 is also known as polymorphism, can significantly impact how our bodies metabolize medications, particularly chemotherapy drugs like irinotecan. So, what happens if someone has a variant in this gene? Certain polymorphism can reduce the enzyme's activity, which leads to a slower duct clearance. This can result in toxic side effects such as severe diarrhea or dangerously low white blood cell count. Now, who does this affect? While UGT1A1 variants are present in all populations, research has shown that the Malay population is underrepresented in pharmacogenomic studies. This knowledge gap can lead to unsafe or suboptimal treatment if genetic differences aren't considered. The impact is more critical in clinical settings, specifically when dosing drugs like irinotecan. Without understanding a patient's UGT1A1 statement, treatment cannot result in harmful side effects. Where this leads us to the final question now. How can this issue be addressed? Well, by identifying and also screening genetic variants in UGT1A1, we can move towards safer, more personalized medicine and also adjusting treatments based on a person's genetic makeup. Now, moving on to the objectives of our study, the primary goal is to detect polymorphism in the UGT1A1 gene among healthy individuals in the population. By doing so, we aim to better understand the genetic variations that might influence drug metabolisms and also treatment outcomes in this specific ethnic group. Our second objective is to design specific primers that target exon 1 of the UGT1A1 gene. This is the part of the gene we focused on our analysis, and the primers are essential for accurately amplifying the region during PCR-based testing. Next, we use high-resolution melting or HRM as our screening method. HRM allows us to detect subtle differences in DNA sequences by analyzing their melting behavior. It is a cost-effective and also efficient way to screen a large number of samples quickly. Once we identify any abnormalities in the HRM curves, we move to our fourth objective now, which is confirming the presence and also identifying the variants using Sanger sequencing. This step ensures that the accuracy of our HRM results by pinpointing the exact genetic changes.
For the methodology, first, collection and data extraction. A white female Oryctus rhinoceros beetle was collected from a pheromone trap in the Solomon Island to obtain a natural and non-laboratory specimen that could represent wild populations of this invasive pest. How we do this, the tissues were taken from the beetle's thorax and legs, avoiding the abdomen to reduce the contamination from gut microbes. And then the DNA was extracted using a customized method involving magnetic beads to isolate high molecular weight of DNA. Then the DNA was further enriched for long fragments, which is more than 10 kb, using a Circulomics short-read eliminator kits. Next, long-read sequencing using Oxford nanopore technology. The extract DNA was used to make 4 sequencing libraries, each run on a minion flow cell. Why we do this? Long-read sequencing is especially good for assembling complex genomes, including repetitive region and large fragment. How we do this? The Oxford nanopore ligation sequencing kit was used to prepare the libraries. The data was base called using Guppy software, which convert raw signal data into DNA sequences. From the data, the team obtained over 37 gigabases of high-quality long-read sequences with an N50 of approximate 11.2 kb. Next, the genome assembly. The genome assembly was carried out using the long-read assembler fly in metagenome mode. Why we do this? Metagenome mode is sensitive to fragment with highly variable coverage, which help to detect and separate contaminant sequences like the mitochondrial genome and Oryctus rhinoceros nudivirus. How we do this? The contaminant circular sequences were identified based on their size and abnormally high coverage and removed. The initial nuclear genome draft had high contiguity, about N50 equals to 7.9 NB. Next, genome polishing with Illumina short reads. What was done? Short read Illumina sequencing data was used to correct error in the long read assembly. Why we do this? ONT data has known errors, especially insertion or deletion in homopolymer region. Short reads are more accurate and can fix this error. How we do this? Illumina reads were aligned to the draft assembly by using BWA-MEM. Pilon was used to correct indels based on the alignment. This polishing step significantly improved gene recovery in the assembly. Completeness went from approximate 65% to 98%. Next, iterative assembly optimization. The multiple version of the genome were assembled using updated tools like Guppy, Fly, with different minimum overlap parameters, 5 kb to 10 kb. Why we do this? To optimize the assembly for highest contiguity and completeness. Okay, how we do this? Illumina reads were used to polish each version. Busco was used to evaluate completeness. The best version had the highest quality metrics. Lastly, gene annotation. The gene were identified using the breaker pipeline, combining prediction with RNA sequence evidence. Why we do this? To accurately located protein coding genes in the genome with real expression data as evidence. How we do this? RNA sequence data from beetle life stages was aligned with his site 2. Repeated element in the genome was soft mask, and then the gene prediction were made using gene mark EX and Augustus. This produced 16,375 protein coding genes and over 20,000 transcripts.
For the results, we start with ONT library preparation and sequencing. We achieve high quantity and quality HMW DNA and yield between 896,000 and 1.4 million raw reads. 2 rounds of analysis was done, and for the second round of analysis, base caller version Guppy V4.2.2 were used to improve the yield of high quality reads. Next, we move to the genome assembly and quality assessment. The figure below shows a flowchart of identification and elimination of organelle, pathogen, and other contaminant genomes, while the figure in the right shows the iterative assembly refinement. Table 1 shows the assembly statistic for Oryctes rhinoceros. The size of the final O rhinoceros nuclear genome assembly is 377.4 MB, which is very similar to the latest assembly for the congeneric beetle or Borbonicus. However, the quality of O rhinoceros assembly is superior than O Borbonicus, both in terms of contiguity and completeness. The original assembly for O Borbonicus was first reported to be 518 MB, but the refinement with the 10 times genomics data led to a 28% reduction in size. The inflated size of the initial assembly was explained as a consequence of an incorrect hybridization of the assembly. The figure shows the Basco comparison to compare with other available nuclear genome assemblies in Coleoptera. 39 out of 41 representative nuclear genome assemblies in the Coleoptera were analyzed and found that one-third were generated with data that included long-read sequences. The mean fragment N50 was 6.9 MB, and the mean Basco completeness was 88.4%. These quality metrics are above the average for a set of 601 assemblies from 20 insect orders. O rhinoceros assembly had the highest assembly accuracy and completeness among 39 benchmark Coleopteran genomes, having only 0.5% missing Bascos and 0.4% fragmented Bascos. A genome assembly from another member of the family, Scarabaeidae, Ontophagus taurus, had the same number of missing Bascos, but twice as many duplicated genes and substantially lower assembly contiguity. The figure shows the RAXML phylogeny with single copy orthologs and sigma GST genes to predict gene model recover to correct phylogenetic placement of O rhinoceros and possible expansion of sigma GST genes in Scarabaeidae. Moving on to the discussion of application of genomic resources for O rhinoceros management, the first one is RNAi target discovery. RNAi can be a promising new approach for insect pest control, particularly for beetles that exhibit a robust environmental and RNAi response. RNAi is a highly specific gene silencing mechanism in which double-stranded RNA directs cleavage of complementary endogenous mRNA. RNAi cause rapid mortality and could be developed into a control tool that is integrated with other pest management tactics. The second is investigating interactions with a biocontrol agent. The assembled and annotated genome of O rhinoceros provides an excellent opportunity to get genome-wide insight into the interaction between this insect pest and its control agent Oryctes rhinoceros nudibvirus.
In conclusion, these studies successfully produced high-quality nuclear genome of coconut rhinoceros beetles, which is the major pest of the palm trees. It was made by using Oxford nanopore technology by using one wild female beetles. This genome is important for future research. For example, like studies about biological control interactions, and also may lead to the pest control development like RNA interference method. However, there is one limitation. Since the genome came from one beetles, it may not show the full genetic diversity of the whole population of beetleids. With these studies, researchers will have better understanding about biology and evolution of beetle, and also it can support researcher as reference to map up biological and pest control strategies for our future and economic. Thank you.